Hello everyone, welcome to the first tutorial on LadyBird. So here I'm going to introduce the application uh, and how to create a project and configure contrast groups. So first, LadyBird is adapted for real-time single-cell data in which you are interested in comparing some groups between them. For example, comparing single cell data from different tissues or comparing normal and abnormal tissues and something like that. So to create a new project, you hit on new project, you name it. For example, I'm going to name it single cell. Your data needs to respect some specific format that you can find here, help, data format, and you find some instructions that you must follow to for your file to be admissible. So you have genes in the columns and cells in rows, and the data file is unique and it must contain all cells of all groups you want to compare. So for example here you must have a contrast column in which a certain character, for example here the letter A, B, C, indicate which groups um, are uh, are displayed. For example here I think I have 10 cells in each of the three groups. We'll see this later in the configuration step. And please know that if your data file contains other columns than gene expression data, it will be ignored and it will not be used in further analysis, at least in the first version of LadyBird. So um, here I want to upload this file and I need to know the data separator. By default, the data separator by LadyBird is taken as semicolon, I think. And if you want to be sure, because if you must specify the correct one. So you open, if you don't know, you open your file in your normal editor. Here I use JEdit, I think. You can use Excel or any other editor, and I can see that indeed the separator is a semicolon. So I use a semicolon, I upload the data, and I will get a yes. Here I get the message unnamed colon were found. So if you have a colon, it must be named. Otherwise, we cannot know which gene it is, and if we cannot know which gene it is, then there is no point in doing this whole analysis. So unnamed colons are dropped out of the data. Here in this case, they are indeed really empty colons. So make sure your unnamed columns are empty or are not relevant to your study before you submit your data. We have our data here. You can see it, verify if everything is alright. If you specify the wrong separator, for example, you'll have different number of columns, or you'll have several warnings that will guide you to choose the correct separator. So now everything is fine. We configure the data. To configure data, you have the small window that pops up. You specify the name of the column. So here you have some instructions in case you don't know what to do. Exact name of column, and by exact name of column I mean name of the contrast column. In this case, it's J. As you can see, the column J distinguishes here between 5 groups from A to E. So where is my figure? Here it is. So I specify J as the name of columns and the name of the contrast column. The number of groups 5 next and here. This is an important step in which you have to name each of your groups and tell tell LadyBird how to detect each group. So, for example, the first group here, the keys A, B, C, etc., and I need to give names for each group. For example, here, let's call them group 1, group 2. It can be anything depending on your project. Here, I'm interested in comparing these 5 groups, and so these names will be used in the tests later on and in visualization plots. I apply. Now I have some warnings. For example, some genes contain non-values in some groups. For example, if your genes are not available in every group, you may get this warning. For example, here these 3 genes were not found in group 5. Do you wish to continue? So if you know that it is the case, then you continue. If you do not, you must verify your data. Yes, so I have no problem, no further warnings. If, for example, you specify a wrong key or a key that leads to a group with very few cells, you may get errors and you need to deal with them. So here you have some information about your groups, the shape of your groups. You must verify that the shape of your groups are correct. Here I have the raw data, the whole groups, and here I have the shape of each of the matrices of each biological group. So 0 unmapped cells, which means that all cells were assigned to some group. I create a project. I have this beautiful window with 2 tabs, one for data visualization and one for statistics. And this will be the subject of our next tutorial. See you next time.